So, in this topic, we will learn about genetic engineering and many terms related to this topic. So, what is genetic engineering? Genetic engineering basically is a process in which a gene is transferred from one organism to another organism to introduce some desired character in the second organism. So, this is called genetic engineering. So, first of all, there is another term which is called vector. We have two examples of vector. One is virus and the other one is bacterial plasmid. So, vector is an intermediate where a gene which need to be transferred is inserted into a vector. We get a gene or a part of gene from one organism by using enzymes, cutting enzymes, and then we introduce that desired gene or desired part of the gene into vector, and then this vector is transferred to another organism. So, this way, by using vector, we transfer desired genes or desired parts of genes from one organism to another organism, and this process is called genetic engineering. Genetic engineering is a process by which we can remove the faulty genes or the faulty parts of the genes by the healthy ones. We can remove faulty parts and we can replace these with the healthy genes. There are some examples. One example is of bacteria. So, genetically modified bacteria are used to produce insulin. So, what is the mechanism? That insulin producing genes are introduced into bacteria. Then these genetically modified bacteria are cultured in a medium, growth medium, and they produce insulin. So, insulin is produced by genetically modified bacteria. And the other important example of genetic engineering is GM crops, genetically modified crops. What is the benefit we can have? That as a result of these crops, we can have improved quality and size of crops, a better quality, improved quality, and bigger size, improved increased yield of the product. Second, we can get crops which are more resistant, naturally resistant to diseases and insects. So, less use of pesticides and the less use of any poisons or any herbicides to kill the pathogens. So, there is another term which is called gene therapy, means treating the faulty genes, and that is also by the genetic engineering. So, gene therapy is used to correct the inherited disorders. We go further for GM crops. So, these are the advantages and some disadvantages of genetic engineering. What are the advantages? That we can have increased yield, we can have the better quantity of the crop, and we can have an engineered crop to contain some nutrients which normally are missing from the food of any segment of population. There is an example of golden rice. Golden rice contains beta carotene. So beta carotene is responsible, if it is eaten in reduced quantity, or the lack of beta carotene in our food can cause blindness. So as a result of genetically modified crops, we can have golden rice which contains the natural quantities of beta carotene to prevent the blindness in people. But there are some disadvantages, that as a result of GM crops, there is a possibility of reduced biodiversity, because the GM crops, they will kill all the other like flowers, other plants in their surroundings. So as a result, the biodiversity of the plants will reduce. Second, there is a possibility that if any herbicide resistant gene is exposed to another weed in the surrounding, so that weed can convert into a super weed. So if any herbicide resistant gene is exposed in the surrounding to other weeds, so these weeds can convert into super weeds, more powerful weeds. And the other disadvantage is that by having the genetically modified crops, there are more chances that the foods can have some more unknown allergies. There is one more related topic for genetic engineering, and that is called adult cell cloning. So cloning is a process by which we produce similar and identical type of cells. And in adult cell cloning, first we get an unfertilized egg, and in the second step we remove the nucleus from the unfertilized egg. So now we just have a egg without any nucleus, no nucleus inside, empty egg, empty egg. Okay, so the nucleus of the egg has been removed. So on the next step we will get a nucleus from adult body cell, not, we will remove the nucleus of the egg and replace it and insert the nucleus from the adult body cell into the empty egg. Now the egg's nucleus has been replaced by the adult body cell nucleus, and then this egg is given an electric shock to make it to divide and convert into an embryo, and this embryo is implanted in a womb of the female, and as a result the clone of that adult is going to be produced. So this is called adult cell cloning. The well famous sheep Dolly was produced by this method of adult cell cloning, but still there is a risk that they can have some health problems, like Dolly had arthritis, but still we are not sure that this was because of this type of cloning, or it was just like by chance. I think that's an all. Thank you very much for watching. See you in the next clip.